All right, good morning, everybody. On the screen up here, we have content that we covered last class. And what we're going to talk about this class is on the next screen. Hello. Thank you. It's 11 o'clock. Let's go. So last class we talked about Mendel's first law. We need to fill in some blanks. Brief review. Mendel's first law is called the law of segregation, which is something we're not supposed to be doing anymore. Segregation is bad. It is. But we have to talk about segregation and how it differs from independent assortment. So in this case, segregation, what is segregating? What's the first blank? This is segregation of something into something. What's being segregated? What would you use to fill in this blank? What word? Alleles. So versions of genes, copies of chromosomes, alleles, are segregating into individual alleles are segregating. They're being divided into what happens at the output of meiosis. You produce the gametes. So Mendel's first law is about the segregation of alleles into gametes. So how chromosomes get divided up into the output of meiosis. Now, if any of you happened to have downloaded this yesterday, please get the version that's on Blackboard now, because I was up early tweaking and adding and deleting and moving around, as I often do. So it's probably a good practice just to download the PDF if you're going to annotate on it right before class. I usually do put it up the evening before. So that was Mendel's first law, how chromosomes get divided up into gametes, segregation of alleles into gametes. Today we do the second law, independent assortment. And so by the end of the class, one of the things I'd like you to be able to do, think about this while we're going. There's going to be the same two blanks. What is Mendel's second law? It's the independent assortment of what into what? So today what we're going to do is talk about the second law. We're going to work on dihybrid crosses. Those are the bigger Punnett squares, not 2x2 Punnett squares, but 4x4 big Punnett squares. 4 rows, 4 columns. One of the goals is to be familiar with some of the common genotype and phenotype ratios, although memorization, of course, is not what we're all about in here. So I just want you to be familiar with them. Don't memorize them necessarily. And the most important thing we do today is talk about why Mendel's second law is really important to geneticists, biologists, plant biologists, doctors, physicians. It's about cross design, about what the possible outcomes of mating between two parents, two plants, two cows, whatever, what do we expect their offspring to have in terms of genotypes? And we'll do a little bit of nomenclature. So here's the first active assignment of the day. Make a little prediction about Mendel's second law. So Mendel's two laws, both of them, have their roots in meiosis, which is why we talk about meiosis first, and then we talk about Mendel's laws. So I've drawn up here a schematic that I'd like everybody to fill in. I'm going to walk you through the first step, and then everybody's going to get released to go do this on their own. And what I'd like you to do by the end of this, about 10 minutes we have in class for this, to fill in this whole chart. Send me a screenshot of this. Attach it to the Google assignment I posted at the start of class today, the one called Mendel and meiosis. So in the next 10 or so minutes, the goal is for you to send me digital versions. It's not for credit, it's participation to help you learn. So we start up here in the upper left-hand corner with a normal diploid nucleus. I'm showing two chromosomes, two different colors, one long chromosome, the blue one, and a shorter chromosome, the red one. So it's diploid. 2 copies of each chromosome. And I've drawn up there one from mom, one M, one from dad, just to give them little labels to remind us that the 2 versions of these chromosomes we got, or this individual got, at fertilization. And I'm diagramming a couple of genes on each of these chromosomes. So chromosome 1 has gene A on it. There are two alleles, a big A and a little a. And the same is true for chromosome 2. It has a gene we're going to call B. It's got one chromosome, one allele, has a big B, one has a little b. Here's a blank to fill in already. What do we call this sort of genotype? If we're talking about two different genes, if it's big A, little a, what do we call that? Is it homozygote or heterozygote? Heterozygote. So big A over little a, one copy of each, two different alleles, it's heterozygous. What do we call it when we're talking about two different genes, A and B, and they're heterozygous at both? This is a blank hybrid genotype. This is a dihybrid. So in case anyone was wondering where that term fits in, that's here. So a monohybrid, when we talk about monohybrid crosses, we're talking about one gene, mono, heterozygote at one gene. A dihybrid cross, then we're talking about two different genes. So that's step one. So the numbers up here at different spots on this figure represent the order in which I'm going to fill in. I'm going to do 1 and 2 with you. Your jobs are going to fill in step 3 and step 4. So if we start with a normal diploid cell in the upper left at step 1, what's the output of meiosis going to look like? Step 2, those last 4 cells at the bottom. If we start with a heterozygote, how many blues and how many red chromosomes are going to be in each one of those 4 cells? One of each. So we start with a diploid, that's the upper left. The bottom row has to be haploid. So there's going to be one of each chromosome. In each of those cells. Haploid. Okay, so that's where we're heading. So now I release you to work by yourselves or in small groups with partners or not. Starting from I think it's easier maybe to work backwards, do it however you like. Maybe easier to go from step 2 to step 3. Given that you know what these are supposed to look like, what will the cells look like in the previous step, before the second division? And from there, what does that mean that this cell has to look like? That vertical dotted line represents where the chromosome is going to divide. So your goal is to arrange, here, let's do post-synthesis. So after synthesis, there are going to be 4 copies of every chromosome. We go from diploid to temporarily tetraploid. So how are you going to arrange those 8 chromosomes in steps 3 and step 4 so that when you do the 2 divisions of meiosis, you wind up getting those bottom 4 cells? So go ahead, raise your hand if you've got any questions. We've got about 10 minutes for this. Hang on just a second.
I have a question.
Okay, go ahead. About 10 minutes. Work on this. Send me a screenshot.
So remember last class we talked about how like the moms and the dads they like separate separately, but if we do this, wouldn't we have like a different phenotype? Because if these are both from the mom, we wouldn't get all of the.
No, these are, so this is from mom and that one's from dad.
Okay, I
And that
thought
one's
the
from
one
mom
M
and
and
that
one
one's
M was okay. And it's also
Attached to Google assignment. There's a Google assignment called Mendel and meiosis. Okay. If you've got a question, do raise your hand.
I think somebody from the last class left this year.
Okay,
So.
thank you. And you have a question?
Oh no.
Okay. Yeah. Okay, that's fine. So you just draw the numbers of the chromosomes and the letters as well. So you've got chromosome 1 distinguish the ones and the twos from each other. That should help. Don't be shy, ask me questions, otherwise I just walk around.
Error, technological
Okay.
issue. Like trying to go to this slide and I don't know, if I try to, I've
weird.
tried opening it.
So this is a PDF, and you're opening it in.
You have
Yeah,
the same app.
right.
And then like I've tried this. I don't know. I've tried opening it up.
So don't say.
I don't know.
Yeah, so if you delete that from, or that,
Yeah, sorry.
The
I've,
other-
like,
no, the other-
tried. I don't know how it's gonna work.
the other option is when you're in explain everything. Sometimes explain everything remembers what you've already loaded. So it maybe delete. That doesn't sound good, but yeah, maybe that's the problem. I'll be right back.
Okay. Sorry.
Let me know if you keep having issues. Hello. Okay.
So for this question, is this what are you on?
Oh, so that's what happens after. So this cell hasn't yet replicated the chromosomes. So after synthesis, what happens? What does that cell look like? And I drew that way. Yeah. So if those 4 cells, if that cell divides there, that means that all 4 red chromosomes are going to go into that. That's something to be worried about. So that's the whole trick, is how do you arrange those 8 chromosomes in that cell so that you get the division you want? Yeah. Yeah, that helps. Yeah. Well, this is the practice period, so we're going to find out if it's right or not. I just want people to try it first. Then we talk about the rules. Yeah. Well, you don't have to do the cross-solution. If you want to email me that, I might not pull it up in class. I might. Make sure it's anonymous. Don't put your name or anything on it. Questions?
Do you restart your other pen?
So how do you line up the chromosomes in step 3 to make sure that every time one of those 2 cells divides, you get a red and a blue in each daughter chromosome?
2 red on each side.
2 blue, 2 red on each side. So So you want, you want 2 blue on the same side? Oh, let's do 3 first. So working backwards from the meiotic products, going up. So there's a blue there, so there has to be, where am I going to draw the red? So imagine that dotted line is the metaphase plate. That's where all the chromosomes line up at meiosis. What did you draw? Where did you put the red chromosome? To the left of the blue chromosome? To the right of the blue chromosome? Above it, below it? So what does this actually look like in the cell when meiosis is happening? The cell divides right there on that dotted line. The chromosomes are going to be paired up across that dotted line, right? So when that dotted line becomes a cleavage furrow and the cell membranes form, you've got a red and a blue on the left side and a red and a blue on the right side. Okay. So that'll look the same over here. Does that make sense? So that everything that's on one side of the dotted line becomes the daughter cell. Okay. So the point of this exercise is worrying about how the chromosomes line up. What's it going to look like in step 4, right before the first division? Let's see. So you've already sent these to me. So we've got...
Not having it.
No, that's okay. You don't have to have sent it to me yet. So let me draw a couple of things that I'm seeing for you here really quickly. So there are a couple of options. You can either do... Let's see. What do we have to do? Reds like that. Does that work? And then what? Blues. So those tetrads, the groups of 4 same color, tetrads of sister chromatids all line up together, side by side. And then 2 wind up going into one daughter cell, 2 in the other daughter cell of each color. So now let's add in really quickly those labels though. This is important. So what are the 2, let's talk about the 2 left red chromosomes. If I put those parental labels back on them, the M's and the D's for which ones came from mom and which ones came from dad, those 2. What labels do we put on these 2? Is it an M and a D? Or is it, how do the chromatids segregate during meiosis 1? Do the 2 chromosomes from mom go together on the left side? Or is it one from mom and one from dad on the left side? It doesn't necessarily mean that the dads always go on the left side, right? That's arbitrary. But it's the 2 copies, the 2 recently synthesized, or the 2 copies of the chromosome from the same parent that segregate together in meiosis 1. This is where independent assortment happens, right now, in the next 15 seconds. Everybody write underneath that on the blue chromosomes what they do. Write the 4 letters. 2 D's, 2 M's. What's going to happen? The red chromosomes are lined up with dad's 2 copies on the left side, mom's 2 copies on the right side. What happens on the bottom? Somebody tell me. Make a prediction.
It would be the same as the red.
So they could be the same as the red. Or, what's the other option? Well, these 2 sister chromatids,
They're synthesized together.
They're-
So you can't separate.
they're synthesized together so you can't separate them.
So the opposite would be like mom and mom on the, with dad and dad, but.
Or, right, the 2 blue chromosomes from mom on the left and the 2 blue chromosomes from dad on the right. So those 2 pairs of blue on the bottom orient themselves irregardless of what's happening with the red chromosomes, without respect for what's happened on the top. Which side dad's chromosomes are on and which side mom's chromosomes are on has no effect at all on what happens to the blue chromosomes there. Separate chromosomes, separate molecules. They line up however they want. Mom on the left, dad on the right. It's arbitrary. In other words, the other thing you could have written is this. You could have had dad, dad, mom, mom, and you could have had the D's over there and the M's over there. Doesn't matter. It's your choice. Well, it's nature's choice. This happens during meiosis. Every time meiosis happens, all of the chromosomes line up right before the first division, right on the metaphase plate, that dotted line. But the orientation, which side mom's copies and which side dad copies are on, doesn't matter.
I was just gonna say, what was the difference?
Right. So this is the difference. Is the orientation.
You forgot to switch them, I think, because the one you drew on the left is the same orientation as the one you drew on the right. Didn't you need to draw the blue chromosomes on the bottom of the
Oh, I'm sorry. Yes. I'm misreading my labeling. Or do that. There, I fixed the problem quickly. So Mendel's second law, what's it called? The law of independent assortment. What's independent about this? What molecules are acting independently of each other? Chromosome 1 and chromosome 2 are arranging themselves independently of each other. So they assort, which is basically another word for segregate. They assort or divide themselves randomly into the gametes. So if we carry that all the way through to the bottom, let's focus on what's drawn here in step 1, the cell that starts meiosis. Which alleles of the 2 chromosomes wind up in the bottom left?
Going based off which one?
So we've got based off of this top cell. So we're gonna carry those the allele numbering, allele lettering down, the D's and the M's.
Dad, mom, dad, mom.
So we've got dads on the left, mom chromosomes on the right. So we've got all dad versions of the alleles in that cell, all mom versions in that cell. Of which chromosome? So it's dad's reds, mom's blues. What happens in the other 2 meiotic products? It's the same or different? Right, so the mom's chromosomes, the red chromosomes are mom's version, and the blue chromosomes are dad's version. So I'll leave it to you to look back on this while you're studying and ask yourself, how would the output of meiosis that we've drawn here, which versions of which chromosomes wind up in the gametes, how does this change if you start with this cell I drew in at the upper right? If you do the 2 divisions of meiosis starting here, what are the gametes going to look like? Can you predict what's going to happen?
Yep, all the dads. Chromosomes on the dads.
Right. So instead of getting gametes that have a mixture of one chromosome that's from dad and one chromosome that's from mom, if both of dad's chromosomes are lined up in the same thing that's going to be a daughter cell on the same side of that first cell, then you get only gametes that have dad's allele of the red chromosome with dad's allele of the blue chromosome. So if you do that, you'll wind up getting the same sets of chromosomes in every gamete, one red, one blue, like we know we should. They're haploid. but you'll get dad allele with dad allele and mom's allele with mom's allele. So this is the essence of independent assortment. If you're working with two genes that are on two separate chromosomes, we're talking about a dihybrid, two genes. How many different haplotypes of meiotic products do we produce? There are 8 germ cells down here. How many different letter combinations are there in those 8? We've got some Ds with Ds. We've got some Ms with Ms. So there are 4. We've got Ds with Ms, Ms with Ds, Ds with Ds, and Ms with Ms.
Wouldn't it become, it'd be all right if they looked at it was 2, because one would be heterozygous and the other one would be homozygous?
So these aren't,
Oh, no.
right, these are 2 different chromosomes. The red chromosomes and the blue chromosomes are different chromosomes. Yep. So we'll have a chance to practice this. So that's the summary. If you're looking at 2 different genes, and independent assortment is happening, you make 4 possible different gametes. How does this impact Punnett squares?
Each gamete has a 25% chance. Let's have 4 of each different one. We have 4 times 4.
So this is why you have to have a Punnett square that's 4 by 4, because every column and every row is what's in one gamete. And if you're talking about 2 genes and 2 alleles, that means there are 4 different types of gametes produced by a parent. So the Punnett square necessarily has to get bigger to accommodate this change. So the independent assortment this time is of, what was assorting this time? We were drawing multiple versions of molecules we call chromosomes. They're genes. But really, when you look at it from meiosis perspective, it's the independent assortment of chromosomes, once again into gametes. So we have to stop thinking about what happens when one chromosome goes through meiosis. Now we're thinking about what happens when 2 independent sets of chromosomes go through meiosis. So what does this actually translate when you actually put together a Punnett square? How does this work? So I gave you this example of a dihybrid cross to perform last week. And you submitted to me a lot of attachments that showed how you would go about making this combination of worms that are rollers with worms that are green fluorescent. So if these 2 sets of genes, the roller gene and the GFP gene, are on different chromosomes, let's assume, how do we set up a Punnett square to know what their offspring would look like? Well, somebody said we've got a 4 by 4 Punnett square this time. So each of those rows and each of those columns has to represent what's in one of the 4 different meiotic products. So let's work on the x-axis first. We deal with the roller worm genotype first. Which genotype of the roller gene, it's minus over minus, which alleles are going to be contributed by a parent that has that genotype? what version of that chromosome goes into every one of these gametes. This individual is homozygous for minus, it has to be minus. So every one of these gametes gets a minus because a parent that has that genotype can only give minus versions of that gene to their offspring. What about GFP? This individual is a heterozygote. There's an asterisk and there's a plus. So how do we divide those among the 4 types of gametes that that individual produces? We've got 4 different types, 4 different columns. How many get asterisks? 2. Doesn't matter which 2. I'll put them next to each other just for fun. Okay, what goes in the other 2 columns? The other allele at that gene, the plus. So a Punnett square, when you're talking about combining 2 different traits or you're working with 2 different genes, you can have those, you have to have a combination in each column and in each row. What is happening at both of those genes? So there should be 2 things, 2 allele designations, 2 letters, 2 numbers, 2 symbols written into each row and each column. Here's where we do a little bit of feedback. A couple of the submissions I got for this Google assignment. How do you combine the roller worms with the GFP? This is what I'm talking about, being creative. So despite the fact that we're in a chapter on transmission genetics, some of you said, well, let's just take the DNA out of the worms and make transgenic worms. We'll take the GFP gene out of one worm and inject it into the other worm. And that's how we'll get the green fluorescence into the rollers. That'd be a perfectly acceptable answer if we weren't in a chapter on transmission genetics, when we're supposed to be crossing individuals together. But that's actually how we make these sorts of combinations of phenotypes, sometimes. Sure, absolutely, that's creative. Make a transgenic organism. What I was looking for was, let's cross organisms together. So correct answers on an exam, I'd give points. We're talking about transmission genetics. So here are a couple of examples of submissions I got that dealt with, let's cross the worm strains together. One that's a roller, one that's green fluorescent. So this is indeed the genotype of the roller worm. It has to be mutant or a minus version of that gene at both chromosomes. And the GFP worms that I gave you to work with were indeed heterozygous. There was 1 asterisk, the green fluorescent gene, and 1 wild type. But let's do a quick group critique. Why are these not all of the possible outcomes? What's happened in this example? What's written here right underneath where I drew in red? Roller worm. Is this worm green fluorescent or not? No. So what's missing from this drawing? What about the genotype that's written here tells me anything about green fluorescence? We're talking about 2 different traits, so we have to talk about 2 different genes in every individual that we draw. So this individual might be homozygous for the roller gene, but for GFP, this worm is probably, this is an assumption that you get to make, probably wild type, plus over plus. both of its copies of its chromosome are normal. It's not green fluorescent, it doesn't have an asterisk. By the same token, what's true over here? Here we're looking at the genotype in the green fluorescent worms. Which gene are we talking about here that's circled in red? Is this the roller gene or is this the GFP gene? It's the GFP gene. What's the genotype of this worm at the other gene, the roller gene?
Heterozygous.
It could be heterozygous. The default assumption is always that if we're not giving you information about a gene, that it's wild type. So this worm at the roller gene locus is wild type. This worm that I gave you to work with, the GFP worm, was normal. It's not a roller, it's just green fluorescent. The other worm, the worm on the left, is green fluorescent. Sorry, the worm on the left is a roller, not green fluorescent. The worm on the right is green fluorescent, not a roller. So I have the same question over here. How would you amend this Punnett square to work with two different genes? What happened in this case? Looks like the genotype at one locus, GFP, went on one axis, and the genotype of the roller went on the other axis. What would I have to add here to make this a 4x4 Punnett square? To accommodate independent assortment, the fact that we're talking about two genes, every one of these gametes, that's the allele, that minus, that's the allele at the roller gene. What is it? Tell me, when you do these questions, what's happening at the other gene as well? What's the GFP allele in that? Is it plus or is it asterisk? Well, is it plus or is it asterisk? Could be either. So that's my question. When you set up a dihybrid Punnett square, it has to be, you have to tell me for every one of those columns and every one of those rows, what's happening at both genes, not just one. So in the end, what's the best way to make a green fluorescent roller? We can even do this without drawing a Punnett square, I bet. So I saw a lot of different suggestions from everybody about how to go about doing this. Some of you said you cross the two strains together, these two at the top. So I've written out their genotypes where I'm showing the roll genotype, shown here. This is a little key. I'm going to do, in parentheses, the genotype with the roller gene first. Then there's a semicolon that separates the genes we're talking about. So this is important formatting for genotypes. You get the two alleles, each separated by a slash. So one version of the chromosome has a minus at the roll gene. The second one it does as well. Those two are separated by a slash. Semicolon, now I'm going to tell you about a second gene. So this worm is plus over plus at the GFP locus. So the minuses in this case, I'm defining, tell us something about what's happening in this individual worm at the roller gene, and the pluses tell us something about the GFP gene. So what happens if we set up a Punnett square to cross these two worms together? you're in Vegas. And you sit down at a slot machine and it gives you these choices. So you've got this worm on the left. It's minus over minus at the roller gene. It's plus over plus at the GFP gene. Those genes independently assort. What version of the roller gene is going to go into each of that individual's gametes? What allele? It's homozygous minus. Every gamete has to have minus. Good. You won that bet. What about the GFP locus?
It's homozygous as well.
Also homozygous, plus over plus. So every gamete coming from that cell is going to have what? Plus. So I better zoom in to give myself more room. So I'd write that semicolon again, because the thing on the left of the semicolon was talking about the roller gene. The thing on the right of the semicolon is going to tell me about the second gene I'm worried about. Is there any difference here between the four columns? Do the four gametes differ at all? No. They're all exactly the same. You get a minus at the roller locus, you get a plus version of the chromosome that contains the GFP gene. So does that really have to be a 4x4 Punnett square? No, it could just be one column. I don't need all of that replication to make things look busy and to make me have more work to do when I fill in all of those squares. If there's only one type of gamete, just make one column. Okay. So that was the first parent on the x-axis, just leaving one column. We know every one of its gametes has to have a minus at the roller gene and a plus at the GFP gene because of its genotype. What happens down the y-axis now on the left? So there's the genotype of the individual in the upper right. So at the roller locus, the first one on the left, what's every gamete going to have? It's going to have a plus sign. Except I can't draw pluses. Okay. And some semicolons to say we're now going to worry about the GFP gene. So how are we going to divide up the genotype of that individual there in the very upper right? It's star over plus. So what am I going to put in this top cell on the left? What do you want me to put? Star or plus? I'll do star. What comes next? Next one down. Let's say plus, okay. Then what has to be true? Well, could be a star. What has to be true now? Plus, right? A diploid individual. Starts out diploid, it's got one star and one plus. After replication, it's got two stars and two pluses. You divide them in these columns however you want. That's how meiosis works. Okay, what was the goal of this? I'm sorry, I totally lost myself in all of this genetics fun. What were we trying to make? We're trying to make worms that are green fluorescent and rollers, and we're going to go to the rave and we're going to sell, that's right. So down here in the bottom, what genotypes are we looking for? Let's list all of the genotypes that we could produce that would make green worms that are rollers. So we've got minus over minus. That makes a worm that's a roller. I defined that when I handed out this assignment. Can't be minus over plus because this is a recessive trait I said at the time. So the worm that's going to be a roller has to have two minuses at that gene. What about at the GFP locus? GFP gene. What possible genotypes make you green fluorescent?
I'll say most asterisk is dominant, right?
Right, it's dominant, so this, plus over asterisk or asterisk over plus, it doesn't matter. What else? There's one more way to be green fluorescent. homozygous for the asterisk. So we're definitely looking for a position in this Punnett square where there are two minuses. Are there any? No. So is this a good cross to make green fluorescent roller worms?
No, you need a heterozygous GFP one.
You're going to need a worm. Yeah, you need a worm that has at least one minus, right? If you're diploid, both of your parents have to give you an allele that you're looking for. If you have to be an F1, an offspring that's minus over minus, you better be sure that each of your parents have at least one minus to contribute.
Oh, I didn't,
Okay, so I'll leave it to you then to decide, once you've made this F1 generation, those are these worms with the genotypes in there when you fill it out, what would you do with them to make green fluorescent roller worms? Okay, finally, last little thing before we go. One of my students, I've told you about Leo's work before briefly. He works on PCR genotyping worms. And he set up this cross between two different strains of worms. One's called AF16 and one's called HK104. So I'm just going to abbreviate them AF and HK, so I don't have to draw the numbers as well. So if he mates an individual that's homozygous AF16 to an individual that's homozygous HK104, 100% of the F1 worms are going to be heterozygous. That's what happens when you cross two pure breeding strains together. So then if he selfs one of those F1 worms, mates it to itself, they're hermaphrodites, right? This is a monohybrid cross like we've done in the past. You can fill in the Punnett square and figure out what are the expected genotypes of the worms in the F2 generation. So that's one thing to do. On the next slide, I'm going to walk you briefly through, so think about doing this, probably on the review guide, study guide for the exam. Here are actual data. So this is what he did. He made those F2 individuals. They've got some sort of combination of AF16 and HK104 versions of genes. What is this? It's a gel electrophoresis. It's an agarose gel. It's polymerase chain reaction products. There's a difference between AF16 and HK104, like we talked about in class when we covered this the first time in class. So there's AF16. It's got a, let's see, which one of these two, AF16 or HK104, has a bigger amplicon, bigger PCR product? Which one's a little bit higher up on the gel, a little bit larger? HK104, the one on the right. Okay. So here's an individual worm in lane 2, one F2 individual, as it explains on the left. What's its genotype? Which versions of the chromosome does it have? It matches, it's a little bit lower on the gel. It matches the AF16 size. So that worm is homozygous. I'm just going to now abbreviate with A's. It's A over A. It's got 2 copies of the chromosome, or AF16. To its right, what's the genotype of that worm? One band, it's a little bit larger. So it's got the HK104 size. It's a little bit larger product. So it's homozygous for HK104, the HK allele, a little bit larger. What's going on here? Two bands, 2 different sizes. That means it's got one chromosome that's AF16 and one chromosome that's from HK104. So that's a het, A over H. So that's the simple way to analyze these gels. You just compare the band size with the standards. Here's your job for the next class, down there in the bottom. This is a different piece of data from Leo, but these are his genotyping data. So go through, just like we just walked through very briefly, I know. Figure out for each of the lanes, what's its genotype? Is it homozygous AF16? Is it heterozygous? Or is it homozygous HK104? Right, so that's the first step. The second step is using that Punnett square that's based on the cross that was used to generate these worms that were genotyped. Predict how many individuals of each genotype we should have seen based on that Punnett square. How many homozygous AF16? How many heterozygotes? How many homozygous HK104s did we expect to see? And then send that to me as an attachment to the other Google assignment that I posted at the start of class. Next time, we've got reading, we've got that Google assignment, and we've got one movie to watch that's on statistics. Good news, you're going to want to hear this, so I know I'm 2 minutes late, I apologize, or maybe more. I won't test on statistics on this exam, I decided. So I don't like to give you, if it's the last thing we covered in class the day before the review session, I like to give you a little bit longer to study that material before I test on it. So we won't do any statistics on the upcoming test. Future tests, yes. In other words, today's information is the last thing that will show up on the exam on Monday. Okay.